all of you. I am Dr. Indira, Associate Professor in Physiology Department in Patil Medical College. Today I am going to tell you the topic that is the erythropoiesis. So what is erythropoiesis? Erythropoiesis, it is a process, it is a formation of RBC. RBC, that is the red blood cells. Formation of the RBC or red blood cells. Before that, you must know what is blood. So blood is a connective tissue. Blood is a connective tissue, and it contains the solid form and the liquid form. Solid form, these are called as form elements, and they contain the RBCs, WBCs and the platelets, and remaining, that is 55% plasma, and remaining this 45% are the form elements, that is RBC, WBC and the platelets. So today we will going to discuss the erythropoiesis, that is related to the RBC, that is red blood cells. So what is erythropoiesis? We will start the main topic, erythropoiesis. So this is the process of production of red cells in the bone marrow, that is called as erythropoiesis. What is erythropoiesis? This is the process of production of red cells in the bone marrow, that is called as erythropoiesis, and it have an infinite lifespan of about 120 days. RBCs, how much lifespan is? 120 days. RBC have lifespan about 120 days, and bone marrow precisely replaces the cells lost by hemorrhage or destruction. Immediately bone marrow replace the red blood cells or destructed cells by hemorrhage or after destruction, and again new red cells are formed. So red cell mass in normal male is about 26 to 32 ml per kg, and in female it is less, about 26 to 39 ml per kg. Why it is less in female? Because male has a more surface area than the female, and that is why red cell mass is less in the female and more in the male. Clear? Then what are the sites and stages of the erythropoiesis? Definition, that is the erythropoiesis is defined as the process of formation of red cells. This is an important component of the hemopoiesis. Hemopoiesis, that is the all blood cells are formed, that is called as hemopoiesis. So there are three stages of erythropoiesis. That is mesoplastic stage, hepatic stage and medullary stage. How many stages of erythropoiesis? That is mesoplastic stage, hepatic stage and the medullary stage. We will discuss one by one. Then before that we will see overall picture of the erythropoiesis. Overall picture of the erythropoiesis. So first primitive cell is hemopoietic stem cell. Hemopoietic stem cell, this is the primitive cell. Then from this hemopoietic stem cell, common myeloid progenitor cell is formed. From this cell, next cell is formed, that is colony forming unit megakaryocyte erythrocyte is formed. Then from this colony forming unit, blast forming erythrocyte cell is formed. Blast forming erythrocyte cell is formed, and from this blast forming cell, colony forming unit erythrocyte cell is formed, and from this cell in the bone marrow pro-normoblast cell is formed, and it is visible under the microscope in the bone marrow. So what are the peculiarities of this pro-normoblast? This is the rounding cell and size is larger, that is 15 to 20 micrometer, and cytoplasm is dark basophilic and central nucleus with slightly clumped chromatin is there, and 2 to 5 nuclei are present in the cell. From this pro-normoblast, second cell is formed, that is called as early normoblast. Early normoblast size is smaller than the pro-normoblast. So size is gone decreasing one by one up to the mature RBCs. Size are gone decreasing. So larger cell, that is pro-neuroblast, then early neuroblast size is decrease here. So basophilic, that is early neuroblast, it is also called as basophilic cell, and it is also round and size is 12 to 18 micrometer. It is less basophilic than the pro-neuroblast, and coarse chromatin nucleoli not seen. Chromatin is present, coarse, and nuclei are not seen. Then next cell that is formed, that is intermediate neuroblast. Intermediate neuroblast. See again, in the intermediate neuroblast size is again get reduced, and here what is this stage is important. Why? Because in this stage hemoglobin starts forming. At this stage hemoglobin starts forming. Intermediate neuroblast, hemoglobin starts forming, and it is also round and size is reduced. It becomes 10 to 15 micrometer, and cytoplasm polychromatic due to the hemoglobination. Hemoglobin is formed, and that is why cytoplasm polychromatic. The nucleus, deep staining nucleus is there, and coarse chromatin material and no nucleoli are there. Then from this intermediate neuroblast, next cell formed, that is the late neuroblast. Next cell formed, that is the late neuroblast. See this size again decrease from this intermediate to late neuroblast, size again decrease, and it is round in shape. Size decrease and goes to 7 to 12 micrometer. It is a well hemoglobinized, that is the hemoglobin well formed and begins to condense and becomes the pycnotic, and eccentric or partially exudate is present. Then from the late neuroblast, next cell formed, that is called as reticulocyte. So reticulocyte, why reticulocyte? Because reticulum is present in this, and that is why this is called as reticulocyte. Cells stain bluish and are larger than the normal red cell. Reticulocytes, these are also called as juvenile RBCs. Reticulocytes, these are called as juvenile RBCs, and up to this reticulocyte, how many days takes place? That is it takes the 7 days to form the reticulocyte. Then from the reticulocyte, mature RBCs are formed. Another 2 days takes place to form the mature RBCs. So total, how many days takes place? Total 9 days takes place to form a mature RBC. So reticulocyte cells stain bluish in color, and reticulum are present. That is remnant of the RNA are present, and that is why they are called as reticulocyte. And these reticulocytes are nucleus is absent, and it is larger than the RBC, mature RBC. So they are called as the juvenile RBCs. Then from this reticulocyte, mature red cell is formed, and this is a normal RBC. It is a biconcave in shape, and biconcave disc shape is biconcave disc, and size is 7 to 8 micrometer in diameter. So this is a mature RBC. In mature RBC, full hemoglobin is formed. There is no nucleus is there, no mitochondria are there. And this is the mature RBC. There is nothing, no nucleus, no mitochondria. Filled with that is these RBCs are live up to 120 days. Okay, so this is a collective or overall picture of the erythropoiesis. Then we will go to the next stage, that is the stages of the erythropoiesis. First stage, that is the mesoblastic stage. Mesoblastic stage, what is that? During the intrauterine life, during the intrauterine life or during the pregnancy, erythropoiesis first takes place in the mesoderm of yolk sac. Erythropoiesis first takes place in the mesoderm of the yolk sac, and mesoderm of the body, yolk sac and the mesoderm of the body, and this is called as mesoblastic stage. This is called as mesoblastic stage, and during this stage erythropoiesis is intramuscular, very early. Erythropoiesis stage 3, that is during this stage erythropoiesis is intramuscular. Then next stage, that is the hepatic stage. Hepatic stage, from the 5th week of gestation. Hepatic stage starts from the 5th week of gestation, and erythropoiesis takes place in the liver and spleen, and that is why this is called as hepatic stage. At that is from the 5th week of gestation, erythropoiesis takes place in the liver and the spleen, and that is why this is called as hepatic stage. Then next stage, that is the medullary stage. Medullary stage means RBCs are formed in the medulla, that is bone marrow. Medullary stage. So from the 5th month of intrauterine life, the bone marrow starts forming red cells, and that is why this is called as medullary stage. However, the erythropoiesis in bone marrow is very slow in second trimester. Erythropoiesis, it starts at the 5th month of intrauterine life, but it is very slow in the second trimester. It is very slow in second trimester, and medullary erythropoiesis become more effective towards the end of the third trimester. Medullary erythropoiesis, it is more effective towards the third trimester, and after birth, bone marrow becomes the main site of the erythropoiesis. After the birth, erythropoiesis is the main site of the erythropoiesis. In the bone marrow, RBCs are formed continuously. Then extramedullary erythropoiesis in postnatal life, it is not formed, or it is absent, or it is called as abnormal. Then extramedullary erythropoiesis in postnatal life is always considered abnormal. So it is present in the bone marrow. Then till adolescent period, bone marrow cavity of all bones are involved in the erythropoiesis, after which erythropoiesis regresses in the limb bones. Initially, all the bones they form the erythropoiesis, but after which erythropoiesis regresses in the limb bones, and after the age of 20 to 30 years, erythropoiesis is mostly limited to sternum, ribs, vertebrae, skull bones, pelvis, and the pectoral girdle. So, after the age of 20 to 30 years, erythropoiesis continues, and the erythropoiesis takes place only sternum, ribs, vertebrae, skull, pelvic girdle, and the pectoral girdle. So, in the adult, after 30 years, erythropoiesis takes place in these bones, all flat bones included. Then, types of erythropoiesis. There are 4 steps. There are 4 major cell stages or stages of erythropoiesis. That is, first stem cell, then progenitor cells, then precursor cells, and the mature cells. Stem cell, progenitor cells, precursor cells, and the mature cells. Then each stage has sub-stages, that is, red cells are formed from the pluripotent hematopoietic stem cells. Red cells mainly form, basically they form from the pluripotent hematopoietic stem cells, and which give rise to committed stem cells. That is, first pluripotent stem cells, then these cells give rise to the committed stem cells, and these committed stem cells form subsequent progenitor and precursor cells. These committed cells subsequent give rise to progenitor and the precursor cells that finally produce reticulocyte and the mature red cells. So these are the steps of formation of the RBC, that is stem cell, progenitor cell, precursor cell, and the mature cell. So one by one cells go on forming, and committed stem cells form the subsequent progenitor and precursor cells that finally produce reticulocyte and the mature red cells in the bone marrow. Clear? Okay. Then in general, during the erythropoiesis, what happens? Following cellular changes takes place. That is, I have seen that is shown you already. That is all the overall picture of the erythropoiesis. Cell size got decreasing, then cytoplasm got decreasing, nucleolus decreasing, then finally there is no nucleus, only red cell hemoglobin is present in the mature adults. So what are the general principles in during the erythropoiesis? That is cell size progressively reduces, then size of the nucleus and number of nucleoli decreases, also chromatin material condenses, and finally nucleus disappears. So finally in mature RBC there is no nucleus, only hemoglobin is there. Then staining reaction of the cytoplasm changes from deep basophilic to polychromatic polychromatic material, and finally to acidophilic type, from polychromatic material to acidophilic type, and this occurs mainly due to the gradual reduction in quantity of RNA material. So this occurs due to the reduction of the quantity of RNA material. So these are the general changes takes place during the erythropoiesis. Then something about the stem cells. What is pluripotent stem cells? What are the pluripotent stem cells? The pluripotent stem cells are the mother stem cells, that is the basic cells. Pluripotent stem cells are the mother stem cells that form stem cells for different cell lines. These pluripotent stem cells, these are the mother stem cells, and that form cells for the different cell lines. And stem cells, including committed stem cells, have two unique properties. That is the self-renewal and differentiation. Stem cell has got two unique properties, that is the self-renewal, and self-renewal means cells are formed from the stem cells, self-renewal and differentiation. So self-renewal and the differentiation, and they have the capacity to renew themselves. Therefore, at number of times bone marrow is depleted of the stem cells. Therefore, number of times bone marrow depleted of the stem cells, and they have the capacity to develop or differentiate. Into the subsequent progenitor cells. So they have this stem cell have the property that is self-renewal and the differentiation, and that is why they have the capacity to develop or differentiate into the subsequent progenitor cell. Then these cells develop from the pluripotent stem cells. These cells develop from the pluripotent stem cells, and they are these committed stem cells. These are developed from the pluripotent stem cells, and these are specific. There are specific two categories of stem cells. That is stem cells from the myeloid cells and stem cells for the lymphoid cells. Then myeloid stem cells form erythroid cells, megakaryocyte, and megakaryocytes, that is the platelet are formed, and monocytic cells and granulocytic cells. Monocytic cells form monocyte, and granulocytic cells form the all granulocyte WBCs, that is neutrophil, basophil, and eosinophils. From these cells, granulocytic cells, all these WBCs are formed, granulocytes are formed. Then erythroid stem cells give rise to progenitor cells. Erythroid stem cells give rise to progenitor cells for erythroid cell lines, and these are two progenitor cells, that is colony forming blast forming unit erythroid and the colony forming unit erythroid. So there are two progenitor cells, that is blast forming unit erythroid and the colony forming unit erythroid. So this is about the committed stem cells. Then there are two types of progenitor cells, that is blast forming unit erythroid and colony forming unit erythroid. From the blast forming unit erythroid and colony forming unit erythroid, they develop from a common progenitor cell, that is colony forming unit megakaryocyte and erythrocyte. Blast forming unit and colony forming unit erythroid, they develop from a common progenitor cell, that is colony forming unit megakaryocyte and erythroid cell. Then this blast forming unit erythroid, these are the burst forming unit erythroid progenitor cell that give rise to the large number of colony forming unit erythroid cells. These blast forming unit erythroid, these are the burst forming erythroid progenitor cells, and these give rise to large number of colony forming erythroid cells. Then colony forming unit erythroid are colony forming units erythroid progenitor cells, and they give rise to moderate number of blast cells of erythrocytic cells. Moderate number of blast cells of the erythrocytic cells, and thus blast forming unit erythroid form colony forming unit, which is in turn forms the blast cells or erythroblast. Colony forming, finally blast forming unit erythroid forms the colony forming unit erythrocyte, and lastly it gives the blast cells, that is the erythroblast. Pro-normoblast, early normoblast, late normoblast. This is the blast forming cells are formed from this colony forming unit erythroid. Then precursor cells. Which are the precursor cells? The blast cells are the first to be morphologically identified cells in the bone marrow. That is pro-normoblast, early normoblast. These are the blast cells, and they first appear in the bone marrow. Under the, we can say under the microscope, in the bone marrow in erythropoiesis, and they are true precursor for the erythrocytic cells. These are the true precursors from the erythrocytic cell, because all the cells, that is all the stages are early normoblast, late normoblast, intermediate, all these form from this precursor. The precursors from erythrocytes are called erythroblast and also called as normoblast. Normoblasts develop from the pro-normoblast cells. These normoblasts develop from the pro-normoblast, and there are the three successive forms of the normoblast. That is early normoblast, that is I have told you, that is early normoblast, then intermediate normoblast and the late normoblast. Intermediate normoblast, that is the important stage. Why? Because in this hemoglobin is start here. Hemoglobin started here, and that is why it is a important stage. Then pro-normoblast. Pro-normoblast, that is, it is also called as pro-erythroblast, are the first blast cell to appear in the bone marrow and the first identifiable cells in the erythrocyte cells. The following are the characteristics of pro-normoblast. That is, this is a large cell having diameter of about the 15 to 20 micrometer, and cell is irregularly rounded or slightly oval. Then cytoplasm is less in amount, occupies about 20% of the cell, and presence of high concentration of the polyribosomes are there. Presence of high concentration of the polyribosomes and makes the cytoplasm intensely and basophilic. So this is about the pro-normoblast. Then second, that is the cytoplasm has high content of RNA. In the pro-normoblast, cytoplasm has high content of RNA, which indicates vigorous protein synthesis in the cell. Cytoplasm contains high content of RNA that indicates the vigorous protein synthesis in the cell is going on. The nucleus is large and occupies 80% of the cell, and it contains multiple nucleoli. In The nucleus is large, occupies 80% of cell, and it contains the multiple nucleoli. And mitosis is present. Hemoglobin is not yet formed in the cell. Then next stage, that is the early normoblast. It is also called as basophilic erythroblast. Then early normoblast or early erythroblast is the first erythroblast to appear in the bone marrow. It has got diameter of the cell is about 12 to 18 micrometer. Cell exhibit active mitosis and cytoplasm is scanty, deep blue and basophilic. The basophilic cytoplasm is due to the continuation of many polyribosomes in it, and therefore early normoblast is also called as basophilic erythroblast. Then nucleus is large and occupies the 3/4 of the cell area. Nucleus is composed of dark violet heterochromatic clumps. Nucleus is composed of dark violet heterochromatic clumps interspersed with pink clumps of euchromatin, and chromatids are connected by linear strands, and this often gives the nucleus that is the appearance of field of the spokes, field spokes. And hemoglobin appears first time in this cell in the erythropoiesis. Then next, that is the intermediate normoblast. It is also called as polyerythromatic erythroblast, and this is also called intermediate erythroblast, and this appears following the mitotic division of the early normoblast. And following are features, that is cell is smaller than the early normoblast and having the diameter of about 10 to 15 micrometer. Cytoplasm changes from blue to pink as hemoglobin dilutes the polyribosomes content. The nucleus is small and occupies about half of the cell area. The distribution of heterochromatin clump in nucleus gives rise to the appearance of the checkerboard pattern, and these are and there are no nucleoli. In the intermediate normoblast, there are no nucleoli. Hemoglobin synthesis increases here during this stage, and which makes the cell acidophilic. Hemoglobin is formed, that is why cell makes the acidophilic, and presence of RNA material makes the cytoplasm eosinophilic. The mixture of acidophilic and hemoglobin and the eosinophilic RNA in cytoplasm that gives rise to polychromatic material. Polychromatic cytoplasm, and therefore intermediate normoblast is also called as polychromatic erythroblast. Intermediate normoblast is also called as polychromatic erythroblast. Then next stage, that is the late normoblast. Next stage, that is the late normoblast. It is also called as orthochromatic erythroblast. This is called as late erythroblast or late normoblast. This is produced by final mitosis in the erythropoietic cells. This is called as final mitosis in the erythropoietic cells, and features are, this is smallest in the erythroblastic cells, 7 to 12 micrometer in diameter, and mitosis is absent. It is smallest in the erythropoietic cells, 7 to 12 micrometer in diameter, and mitosis is absent here in this. Then cytoplasm is deeply eosinophilic, giving the appearance of an orthochromatic cell. Cytoplasm is deeply eosinophilic, giving the appearance of orthochromatic cell. Hence, this is also called as orthochromatic erythroblast. Late normoblast, it is also called as orthochromatic erythroblast. Then nucleus is small and pycnotic at the beginning. Then sometimes dark nuclear chromatin material are arranged in a typical pattern, give rise to the appearance of cart film, and finally pycnotic nucleus disintegrates. the hemoglobin synthesis increases and almost complete in this state. Hemoglobin synthesis increases and almost complete formation of the hemoglobin in this stage, that is the late normoblast. Then reticulocyte. Reticulocytes, up to this formation of the reticulocytes, 7 days takes place, and these reticulocytes are the immediate precursor of red cells. Therefore, they are also called as juvenile red cells. Reticulocytes, these are also called as juvenile red cells or juvenile RBCs. They are mature cells with full complement of hemoglobin. They are the mature cells with full complement of the hemoglobin, and they are slightly larger than the red cells. They are slightly larger than the red cells. They have network of reticular nuclear material, and the reticular network is nothing but the remnants of the disintegrated organelles and especially from the nuclear fragments. This reticular network is disintegrated organelles and especially from the nuclear fragments, and due to the presence of reticular network, the cells are called as reticulocyte. Due to the presence of reticular network, cells are called as reticulocytes. Then reticulum is stained by suprametal stain. Reticulocyte, if you do the practical, that is the separate demonstration is there. Separate demonstration is there, that is of the reticulocyte. So reticulum, they stain with the special stain that is called as the suprametal stain, that is methylene blue and the chrysine blue. These are the stains to stain the reticular material of the reticulocyte. So, and this is called as suprametal stain. The hemoglobin synthesis continues to some extent in some reticulocytes, and the reticulocyte count is very less. Reticulocyte count is very less. It is about 0 to 1%. Reticulocyte count is very less. It is about 0 to 1% of the red cells in the adult. And erythrocytes, that is, erythrocytes are the final cells in the erythropoiesis. Erythrocytes are the RBCs, are the final cells in the erythropoiesis. They are the biconcave discs. They are like a biconcave disc, and they have the diameter of about 7.4 to 7.5 micrometer. The duration of the erythropoiesis, how much duration takes place. So the total period of erythropoiesis occur in 7 to 9 days. Total period of the erythropoiesis occur 7 to 9 days. It takes 5 to 7 days for the progenitor cells to become a reticulocyte. So up to the formation of reticulocyte, it takes 5 to 7 days, and another 2 days for reticulocyte to become a mature red cells. Another 2 days for reticulocyte to become a mature red cell. So total period of erythropoiesis is that is the 9 days. Within 9 days, to mature RBCs are formed. Then regulation of the erythropoiesis. How the erythropoiesis regulation takes place? There are many factors that effectively control the erythropoiesis. There are many factors that effectively control the erythropoiesis. In physiological conditions, total cell mass is maintained at its optimal size by the suitable adjustment in the rate of erythropoiesis. There are many factors that effectively control the erythropoiesis. In physiological condition, total cell mass is maintained in its optimal size by the suitable adjustment in the rate of erythropoiesis or formation of the erythropoiesis. Then there are so many factors that control the erythropoiesis. There are so many factors that control the erythropoiesis. That is, these are which are the factors? Hormonal factors, then dietary factors, and the other factors. The hormonal factors, which are the hormonal factors that contain, that is the erythropoietin. Erythropoietin, what is erythropoietin? It is a hormone secreted from the kidney, secreted from the kidney, the cells of the kidney, and androgens, estrogen and thyroxine, and the anterior pituitary hormones. Which are the anterior pituitary hormones? That is growth hormone, main growth hormone. Then other hormones are TSH, thyroid stimulating hormone, adrenocorticotropic hormone, then luteinizing hormone, then follicle stimulating hormone, and prolactin. These are the anterior pituitary hormone which control the erythropoiesis. Main, that is the erythropoietin. Erythropoietin, that hormone secreted from the kidney. Then androgens, estrogen, thyroxine, and anterior pituitary hormone, that is growth hormone, TSH, ACTH, LH, FSH, and prolactin. All these hormones control the erythropoiesis. Then corticosteroid also they control the erythropoiesis. Then interleukins. Interleukins, they are also important for the control of the erythropoiesis. Then dietary factors. Which are the dietary factors? That is mainly vitamins. Which vitamins? That is vitamin B12, vitamin C, and folic acid. These are the main factors for the maturation of the RBCs. These are the main factor for the which takes place for the maturation of the RBCs. Vitamin B12, folic acid, and the vitamin C. Then proteins. Proteins also required, or they are the important factor for formation of the RBCs. Then proteins, then minerals. Which minerals? That is iron, copper, cobalt, and nickel. Minerals, iron, copper, cobalt, and the nickel. Then other factors include the intrinsic factor, chemical factor, and environmental factor. That is environmental factor, it includes the high altitude, that is hypoxia. At high altitude, environmental factor, that is at high altitude hypoxia is there. So in that condition, erythropoietin is secreted more, and that leads to erythropoiesis. And drugs, these also stimulate the erythropoiesis. So these are the factors, that is hormonal factor which regulate the erythropoiesis, hormonal factor, dietary factor, and other factors. So hormonal are important. Hormonal contains the erythropoietin, androgen, estrogen, thyroxine, anterior pituitary hormone, corticosteroid, and interleukin. Dietary factor, vitamin B12, folic acid, and the vitamin C. Proteins, mineral, which mineral? That is iron, copper, cobalt, and nickel. These are also required for the formation of the RBCs. And other factors, that is intrinsic factor, chemical factor. Thank you.